Thank you, Chelsea, for that lovely introduction and for inviting me. I'm really happy to be here. I gave the first iteration of this talk at MONOCOTSvii, and I'm really happy to report that a lot of what I presented in that was probably very wrong. So I'm really happy to give an opportunity and give an update on something that I think is a little bit more accurate. What you're seeing here on the screen is a laser ablation tomography. So we're taking a serial section through an umbellate inflorescence. And I won't be talking about these data, but I think it's really, really cool because it shows the complexity of these really high condensed structures. I want to start this talk by discussing this kind of broader question in evolutionary biology, in particular, why evolutionary models are important for our interpretation of evolutionary biology. And to kind of drive this point home, I'm going to walk through this example from Church and Extrover, but I've replaced their arthropods with some plants. And in a lot of comparative biology at macroevolutionary scale, we're often presented with this type of data, where we have a phylogeny that represents the relationships amongst our organisms, and we have some comparative trait data. In this case, the expression pattern of some transcription factor. And in two of these, in Calochortus and Thalictrum, let's say, we have this transcription factor expressed at some developmental stage 1, but in Aquilegia, it's expressed at some developmental stage 2. So as macroevolutionary biologists, we might see this pattern and be like, that's really interesting. Perhaps this change in expression from developmental stage 1 to developmental stage 2 led to some really interesting changes that may be adaptive, maybe some other conjecture that we can come up with and kind of expand our understanding of these processes. But in order to do that, we need to make an inference about the expression pattern in the ancestor of these taxa. And in order to do that, we need a model of evolution. And here I've depicted this model as a cartoon, where I'm simply representing the expression, this model is expressing the evolution of this expression pattern, where it goes from developmental stage 1 to developmental stage 2. And we can fit this model to our data, and we get this result, this ancestral state reconstruction, and we can see that, similar to our conjecture that we just described, this changed expression stage 2 evolved in Aquilegia. But what if we fit a different model, one that allows for reversibility? If we fit this model, we might get a different interpretation of biology, one that is meaningfully different from this other one. Here we see that, in fact, it wasn't a de novo evolution, but in fact, it was a reversal from ancestral condition. Now, of course, this is a dummy example, right, just to drive this point home, but it really highlights that models are important for our interpretation. And more importantly, I want to pose this question of how do we choose? So I depicted these as little cartoons, but these are statistical approaches. And on the one hand, we can use statistical justification, such as different criterias, information theory, things of that sort. But I think as empirical biologists, we have more to contribute than just our data. We have deep understanding of our systems and the relationships and these patterns. And there might be some information that's not formalized into these methods that we can then make the decision a priority to choose amongst these models. So I want you to keep that in the back of your mind throughout this talk. It'll be kind of the focus of this question. But now to transition to the trait that I'm studying inflorescences. I think I don't need to explain to this group of audience how cool inflorescences are and how diverse they are, but I'll just give you a brief kind of introduction. I think botanists for a long time have acknowledged that these structures, which are different from the flowers sensu stricto and are different from the vegetative portion of the plant, are really diverse across angiosperms. Just to kind of give a couple examples, I like this definition of inflorescence by Troll, the shoot system which serves for the formation of flowers and which is modified accordingly. We all know the classic sunflower example, that's a misnomer, where we have an inflorescence that's converged upon a flower, but the opposite is known to happen. For us, plant taxonomists, we can quickly realize this is an iris, a single flower that has these interesting petaloid carpellate features, but to a bee, that may not have taken a plant taxonomy class, they might misinterpret this as one of 3 flowers. So this here resembling a single flower that the bee can enter. And there's a lot of morphological features that have allowed this to happen, including like the presence of these interesting UV spots. So in the same way that an inflorescence can converge upon a flower, there's also evidence of a flower converging upon an inflorescence, highlighting the importance of these structures. They can do other really cool things. In Calamus, they're armed. Some of the palms are used for climbing. We heard a little bit about this yesterday in Trianthia. The stem of the inflorescence forms these glandular structures that are involved in carnivory. But they're also important for facilitating outcrossing and inhibiting selfing. So we may be familiar with dichogamy, the separation of carpellate and staminate features on an individual flower, but in some instances you get this thing called synchronous dichogamy, where the entire inflorescence is either in a carpellate phase or a staminate phase. And this is thought to be involved in helping promoting outcrossing. And even though inflorescences are really important for evolutionary biology, and I think we all recognize their diversity, we lack an understanding of their organismal homology. This is an image from Stevens from 1973, who kind of first conjectured upon how you get different kind of place efflorescences and kind of a modern day understanding of evolutionary biology, and conjectured about how you transition between different inflorescence types. But this model hasn't really been tested in an evolutionary framework, and I think this really it's a really key piece of this broader plant evolutionary picture. And I think as a botanist, I hope that that's sufficient justification for this work, but I often speak with non-botanists, and I think if we lack an understanding of the organismal homology, we also lack an understanding of the genetic homology. And that comes really important to play when we think about crops. We have the cob of a corn, the vine of a tomato, the bunch of bananas. These are all inflorescences. Within these respective fields, there's a bunch of plant breeders and trying to dissect the genetic basis of these functions, but there's often no crosstalks. So hopefully I've justified the importance of this trait. But the specific inflorescence that I'm studying is the umbel. Umbel or umbellate inflorescences are those where the flowers seemingly arise from a single point. Here I have a couple of interesting examples. And what motivated my dissertation was these hypotheses for how you evolve an umbel. In particular, there was this hypothesis in the literature that in order to get an umbellate structure, properly termed as a bosterix, which is found in the family Amaryllidaceae, you had to go through the sequential branch rearrangement followed by differential branch elongation, loss of the terminal flower to get to this structure. But there's a competing hypothesis in other taxa that it simply occurs by differential branch elongation. And this first hypothesis is rather complex, but in part it was supported by developmental morphology. So early studies of the young inflorescences of families in the Amaryllidaceae had flowers that initiated in a sequence that was consistent with extant taxa that had this kind of thyrsoid, or in this case, a dichasium. So the first part that, so really my dissertation was trying to test this hypothesis. Is one or the other important in, well, the true or is kind of the basis of umbellate inflorescences, or are there other pathways for which you can get an umbel? So the first part of this work is published, but I just want to briefly touch on it, kind of give some context. Essentially what we did is we did a developmental morphological survey of different umbellate structures across the monocot. So here in red are different instances where umbellate structures have evolved. We grew these plants up or collected them in the field in the greenhouse, and we did a time series, so studying their inflorescence ontogeny throughout time. And this was with the help of 3 students, Aaliyah Holliday, Tara Lurie, and Jason Rose. And we also studied some taxa without umbels. To cut to the chase, we found that umbels evolved via convergent evolution at least 4 different ways. Here I'm showing you representative micrographs of the different types. We have umbellate inflorescences that arose from racemic origins, where you have the primary axis and the lateral axes are simply flowers. And we have these 3 instances where umbellate inflorescences are arising from some form of thyrse or thyrsoid structure. And really interestingly, within these 2 cases, there was an additional cases of parallel evolution. So umbels are evolving via the same genetic mechanism. So this in general highlighted the importance of this inflorescent type in the evolution of monocots, given that there's so many different ways to get there. And another way of looking at these data is to kind of elongate them out into where we kind of exaggerate the stems to highlight that we have kind of racemic origin, and then we have this thyrse with lateral bostrices, thyrsoid with dichasia, and then thyrsoid with cincinae. So I've shown you this. This really highlighted that there's different ways to make an umbel through developmental time, right? But this hasn't tested the hypothesis as over evolutionary time. Are you getting umbellate structures kind of from the respective kind of types? And for that, we need to really make an inference of the ancestral condition of an umbel. Because we might get a situation where yes, we see racemes. They're kind of simply doing this branch elongation, and we're seeing that thyrsoids are doing this kind of weird thing to get to this umbellate structure as was depicted in these hypotheses. But we might also get something a little more complex, right? What if all umbels arose from the racemes? It sounds ridiculous, but in order to test this, we need an evolutionary framework. So to do that, kind of going back to the original introduction of this talk, we need to collect comparative data, we need a phylogenetic framework, and we need a model of evolution. For the comparative data, myself and two aforementioned students went through and collected inflorescent types from the literature from across the monocots, trying to sample as much diversity as we could. So approximately 2,500 species is roughly where we ended up with. And essentially what we tried to do, if any of you have studied inflorescences, you've likely been confused at one point or another, in part because inflorescence classification is authoritative and it's based off of different people's authorities. And so confusion often arises when you have contradicting authorities or different people are not specifying what authority they're using and essentially clearly defining their definitions. So in general, I used Endros 2010's classification scheme, which I've kind of diagrammed out here. It doesn't account for all kind of really nice variants, but it accounts for the large majority of inflorescence types for this type of macroevolutionary scale. And then for the phylogeny, we use this large kind of super tree that was generated in 2019 of the monocots by Howard et al., in part because it could accommodate all the tips that we included, and it largely represents our understanding of monocot evolution, although there are some interesting things that we've heard about during the symposia. I do want to make sure everyone understands that our inference is only as good as our data, and if our data is inaccurate or there's contention there, that will be reflected in our inference. So keep that in the back of your mind. And then the last part, we need a model of morphological evolution. So I want to spend a little bit of time talking about this. So for discrete characters, if anyone's ever done phylogenetic comparative method, the models of discrete characters are all based off these continuous time Markov chains. And these were originally developed to study the substitutions of nucleotides. Modern day understanding of phylogenetics is based off of this model. And the way it works is you have these discrete states, which in this case are the nucleotides, and you have transition probabilities that represent their evolution in the same way that we had these for that dummy example I proposed at the beginning. And in 2001 and early 90s, people realized, well, we can use these models to study more than just nucleotides. We can study any discrete state. So we could, since we have discrete classification of these inflorescences, we could simply use an extension of this model to study these. However, the CTMC makes some unrealistic assumptions, particularly about inflorescences. The first is that inflorescences fundamentally not discrete, right? This contention in classification is partially based off that they are not discrete characters, they're fundamentally continuous, they vary in their branch lengths, the number of flowers, etc. And the other one's a little bit more nuanced, but these types of models are said to be memoryless. Your past state does not influence your future state, kind of a philosophical thing. But basically what it means is that if you've been, let's say, over here for a really long time, that history doesn't really matter. All that matters for your potential evolvability is where you could go. And this might not make sense for a character that is a developmental morphological character, because we have this notion that the longer you're in a particular state, the harder it is to evolve out of this state. And we see that pattern across inflorescences. They're generally, the architecture is generally fixed at higher taxonomic scales. We don't often see taxa with polymorphisms kind of at the species level. So what can we do? We turned towards the developmental biology literature. In developmental biology literature, there's these other model called the transient model. I know a lot of different models. They all got fun names. But this model is not an evolutionary model. It's a model of morphogenesis. So it aims to simulate inflorescences based off the developmental principles of the repeating growth of plant structures. And it was able to generate this morphospace that was able to simulate a wide diversity of inflorescences. Not all of them, but some of them. The way it works is it models this factor called vegetativeness that decreases as a function of time and is a property of the stem. So here we have this model simulating a raceme, and you can see that vegetativeness is decreasing through time as this inflorescence grows, and when it reaches this flower threshold, it terminates into a flower. And what the authors notice is that the interplay of vegetativeness between the primary axis and the lateral axis really dictated the architecture of the inflorescence. So in this case, the primary axis reaches the threshold after this orange line here, which is the first lateral axis. In contrast, you can get something like a dicasium if the primary axis reaches the lateral axis if the primary axis reaches this threshold before the lateral axes. And so you can get a nice diversity of inflorescence architectures. But moreover, what the authors found is that this seems to represent, at the time, our understanding of the genetics that underlie inflorescences. So this structure right here on the morphospace is the Arabidopsis double raceme. And by knocking down leafy, one of these important genes involved in inflorescences, you get a structure that's more highly branched. So it's like moving through the morphospace this way. And by knocking down TFL, you get an inflorescence that's less highly branched, like moving through the inflorescence this way. So it's kind of a nice way of accounting for the generative process of inflorescence diversity. So how can we use this in kind of an evolutionary sense? Well, what I kind of thought is that we could take our discretely mapped states, or discretely scored straits, and map them onto this morphospace, therefore kind of changing their rescoring them by a set of continuous parameters that the morphospace uses to generate these structures. So you can see here a swath of different inflorescence types. And so what I'll stop here to say is that not all inflorescence diversity, as I said, was one of the limitations of this approach, was represented in this morphospace. So we had to do a couple of things in model testing with reversible jump MCMC to test these hypotheses. But you were able to generate a wide diversity of inflorescence architectures. And because you've essentially rescored your discrete states into pairs of continuous traits, that opens up the realm to models of evolution that account for continuous evolution, mainly the Brownian motion types of models. So here we have a Brownian motion simulation on a phylogeny. On the y-axis, we have the value of the trait, and we have some trait evolving through time. And you can see that with Brownian motion, we have this lineage that's gone off into like green space. For it to get back up to any of these spaces, they would take a little bit of time. So it seems to account for this notion of difficulty of evolving, or rather transitioning between different discrete states. And so what we can do is fit both of these models. We can compare the threshold model and these continuous time Markov models, specifically in attempting to infer the ancestral condition of umbellate inflorescences in the monocots. And what I found is that the continuous time Markov model seemingly consistently infers a racemic origin of various umbels, in contrast to the threshold model, or excuse me, the threshold model is a flavor of these Brownian motion type models, but we call it the threshold model. So many models. The threshold model is able to more consistently infer these diverse origins of inflorescence architectures that I showed in the first section. And what I really want to highlight is that this approach didn't actually incorporate any of the developmental morphology from the first section. So this is only based off of the kind of mature inflorescence architecture. So in some way, this model, I argue, is much more realistic. And so this kind of gets back to this point of how do we choose between these models? Well, I would argue that the threshold model, because it's more realistic, it's accounting for these processes that we know as kind of organismal biologists are important for the evolution, is more realistic than the continuous time Markov models, which are simply kind of the default models that people use when inferring and studying discrete states. So if we think about this broader question of the origin and development of umbels, I showed you that they're important inflorescence types, at least in the monocots. They've evolved via multiple different mechanisms, and I've provided a kind of a framework of incorporating developmental genetics and our understanding of the organismal properties that produces phenotype into evolutionary models. But of course, there are limitations. So at the moment, we haven't accounted for the uncertainty in the phylogeny. So the phylogeny is, as I mentioned before, there's seemingly different, in large in part consistent, but there's very important areas that have different topologies, and so accounting for that is necessary. Luckily, it is relatively straightforward. It's something that I'm working on now, and there's a statistical framework for doing so. But also, we need to build more models that more accurately represent the diversity of inflorescences. Particularly, there's a lot of work in the grasses, specifically corn, in order to understand the factors that are involved in inflorescence development, and kind of generating morphospaces for these taxa will be important in the development of these methods. So with that, I'd like to acknowledge the Spec Lab, where I did all of this work and the support of Chelsea Specht. My funding sources, my collaborator, Will Freeman, who helped me with the statistical approaches, and of course, the student co-authors who are helping out with this work. And thank you, the audience. I'd be happy to take any questions.
Thank you so much. Time for questions.
Hi, Jesus. This is really exciting what you do. I'd like to know, you're talking about discrete genes, but could you introduce some sort of networks or like relationships between like genes in these models?
So yeah, I think the kind of the general principle of these models is to, or as these models were originally proposed, the developmental genetics models is like, what is the least amount of data that we can use to generate all this diversity? Because obviously there's going to be idiosyncrasies within every taxa, gene duplicate, like there's taxa-specific things that are happening that are important for those taxa. But the approach here was more broad to generate these methods that kind of accounted for large parts of diversity. So it's this trade-off between specificity and kind of generalization. I think there's definitely room to develop like taxa-specific models that would account for these processes like different crosstalks and different genetic regulations that are specific to the taxa. Yeah, but and they can definitely be incorporated in this framework. Yeah, good question.
Any other question?
Hi, that was such a fascinating talk. Do you think that it's more difficult to move between certain regions of morphospace? Like, is it difficult to transition from an umbellate inflorescence to a different kind of inflorescence?
Yeah, that's something that I am currently working on. And if you just look at kind of the distributions of inflorescences across monocots, you do see that you have like large families in some cases that are kind of stuck in one space. And so it seems to be kind of taxa by taxa. But the fact that these structures have evolved multiple times indicates that if you give it enough time, you seemingly can evolve between certain things. There are some transitions that we don't observe, though, and I didn't have too much time to talk about here, but there are some cases where, at least in the monocots, we don't observe these, and this could be simply because it hasn't existed yet, or it could be some kind of more deeper kind of developmental constraint that might be inhibiting that transition. Yeah, good question.
Yeah, thank you for your talk. So the transient model proceeds from the leafy terminal flower system in Arabidopsis. But we know that, for example, in Petunia or Gerbera, there are other regulation systems. So do you think it is appropriate to derive all inflorescences from the Arabidopsis model?
Yeah, that's a great question. And no, it's not, right? And I think inflorescence, this kind of goes back to this broader topic that inflorescences, unlike other traits in plant biology, like the ABC model, right, where there's seemingly clear orthology in these genes and they more or less function in floral identity, in the inflorescences, there doesn't seem to be a set of core genes that are conserved. And the answer is why. I think a lot of your work, for example, on fumes is really interesting because it highlights that potentially there might be some kind of floral identity genes that are involved in this. But what I like about this approach and why I think it's really important for the field of evo-devo is that this approach is explicitly accounting for some notion of organismal biology in models of evolution. A lot of times models of evolution don't think about the phenotype themselves or how that phenotype is evolving. So I think there will be trade-offs in specifying these models, whether kind of at broader scales or within lineages that are specific. But I agree that this model is very inadequate in the sense that it is solely derived on one model system. So we need more comparative work. Good question.
Thank you so much. We don't have more time, so thank you, Jesus.
Thank you.